So the next speaker will be Anu Meerwaldt. She is also a PhD student in the Nanomedicine lab, and she will talk about the use of fluorine MRI to study myeloid cell dynamics.
Okay, thank you for this opportunity to present. I'm very excited to talk about our project where we developed this new nanobiologic platform for tracking myeloid cells in vivo. So in cardiovascular disease, inflammation and its failure to resolve are one of the key underlying factors contributing to cardiovascular disease and also its complications. And one of the hallmarks of this disease is the increased myeloid cell turnover in the hematopoietic organs, and then these cells are then being recruited to the sites of inflammation and injury. And the current techniques to investigate these processes are based on snapshot techniques such as flow cytometry. So we really sought out to investigate whether we could find a way to actually track these processes over an extended period of time in vivo. So for this, we developed this highly innovative nanobiologic platform, which is also an HDL platform, as Mandy mentioned. So it consists of a phospholipid and ApoA1 protein corona, and the payload are perfluorocarbons. And this payload makes it possible that it can be detected with fluorine MRI. And the excellent thing about fluorine MRI is that there isn't any endogenous signal in the body, so it's truly a nice hotspot technique. Additionally, we can label these nanobiologics with radioisotopes or fluorescent dyes so that we can do also multimodal imaging and tracking. So the concept of study was to investigate our platform in the case of cardiovascular disease. So what happens, we injected these nanobiologics into a mouse, and then they are taken up by the inflammatory immune cells in the hematopoietic organs. And when needed, then these cells are being recruited to the site of inflammation and injury, so for example to the aorta and to the infarcted myocardium. We started this project, the in vivo part of this project, by screening 3 different sizes of these nanobiologic platforms. And here you see depicted a fluorine MRI overlaid to a proton MR image, and this is 48 hours after injecting to a mice. And we could clearly appreciate that there is an highest uptake in the hematopoietic organs, in the spleen and in the bone marrow, with the largest nanobiologic formulation. And we also imaged them over several days, up to 28 days, and the signal stayed more or less stable. Additionally, we investigated the cellular uptake with flow cytometry, where we could see the similar effect that the largest particle is taken up the most. We validated this result with these radio-labeled nanobiologics, where we could also see a nice signal in the spleen and in the bone marrow with PET-CT. We then moved to investigate whether our technology has actually some use in the context of cardiovascular disease. And for this we used ApoE knockout mice that when combined with a Western diet, they will develop atherosclerosis over a period of time. And we wanted to see how our platform works both in the context of ischemic heart disease as well as in atheroprogression. So first, in the context of ischemic heart disease, we kept these mice 6 weeks on a Western diet, so they were in an intermediate stage of atheroprogression. We injected them with our nanobiologics, and 48 hours after, we gave them myocardial infarction, and then we did imaging. Here you can see a control mouse that has been injected and imaged 3 days after injection, and you can clearly see the nice hotspots in the spleen, in the liver, and in the spine. Now, what happens when we take mice that have given myocardial infarction? We see this dramatic depletion of the fluorine signal. both in the spleen and in the bone marrow. So this was, of course, very exciting. And the next question is that what happens with this signal? What happens with the cells that taken up our nanobiologics, and whether we can actually track them further? And we did this with these radio-labeled nanobiologics, and we did autoradiography. These are slices of hearts, and we could clearly see these hot spots in the infarcted myocardium, which show that our nanobiologics are taken up by the heart tissue. And we corroborated this result with flow cytometry, where we see that there's a massive influx of these pro-inflammatory monocytes in the infarcted myocardium at the same time point. So here we can really appreciate this very robust effect that we can track these myeloid cells in vivo. Now, of course, the underlying cause of ischemic heart disease is atherosclerosis. So we wanted to investigate whether we could also use our technology to see the more subtle changes that are introduced in atheroprogression. And here we had 3 different groups of mice that were on various stages of atheroprogression. We injected them with our nanobiologics, and we spent a lot of quality time with the preclinical scanners, scanning them up to 28 days. And we saw more or less stable signal over time, except we were able to discern this one sweet spot that was in the intermediate stage of atheroprogression. And then we saw a diminished spleen signal. And this was, of course, very interesting. And it coincides very well with the highest myeloid cell turnover in the spleen that is introduced at this point of atheroprogression. And we were also able to show that there is actually an increased accumulation and recruitment of pro-inflammatory cells in these atherosclerotic lesions at the same time where we see this signal diminishing. So to conclude, we developed this very nice nanobiological platform. We tested it both in the context of ischemic heart disease, where we saw very robust effect, but also we were able to see some subtle changes in the case of atheroprogression. And we believe that this technique can also be very useful in the context of other inflammatory diseases. So we are also looking into other applications, and for example, I'm currently investigating whether we can see similar effects in the case of stroke. I would like to conclude acknowledging, of course, the people in the Mulder lab, all our collaborators within and outside of TMII. Thank you.
Any questions?
So after the after the fluorine gets taken up by the myeloid cells, what is its fate in those cells over several days?
That is an excellent question, because of course you could argue that maybe it just sort of the cells die, or the fluorine is just deposited to the organs. So we haven't really looked at it at a cellular level yet, but because we could see this ingress from the hematopoietic organs that basically store these myeloid cells, we believe that it's really retained by the cells for an extended period of time.
Have you looked to see if the particles are staying intact in the cell?
No, we haven't done that, no.
Other questions? Okay, then we end the session with the speaker.
Thank you for